Hello everyone, my name is Mandaogade. I am an assistant professor in Department of Biotechnology from Tulsiramji Gaikwad Patil College of Engineering and Technology. So today we are going to learn subject Bioprocess Engineering and our topic is Introduction to Fermentation Process. So in our day-to-day life we are all know that many fast foods are made because of the fermentation process just like idli, dokla and dosa. So what is fermentation process? So we are moving for first of all for the definition of fermentation. So definition of fermentation. Fermentation is a metabolic process in which microorganisms just like bacteria, yeast, fungi or actinomycetes, all of these are convert complex organic substrates, just mainly carbohydrates, into simpler compounds such as alcohols, organic acids, gases, enzymes, antibiotics or biomass. As we all know, humans need food for their regular practices or their regular activity. So after taking up our food, these foods are going to our digestion process, and after the digestion, all of the nutrients just like sugars, proteins and vitamins go to for their respective works and respective places in all of the body. So just like our human body, microorganisms do the same after taking up nutrient from different sources. Just like we are taking an example, nutrient media. In nutrient media, different types of components are there: beef extract, peptone. So when we are growing microorganisms on the nutrient media, microorganisms take up that nutrient and do their metabolic activity, and by their metabolic activity different types of product formation are there. Just like all of you know, the alcohols, organic acids, gases, enzymes, and by the metabolic activity just like central dogma, transcription, translation, and there in the end after the formation of protein is there. Just like different types of these are the metabolic process, and for this metabolic process all of the microorganisms require organic substrate. So main all organic substrate is the mainly carbohydrates. So different types of carbohydrates is needed, just like glucose, fructose and sucrose. So the term fermentation comes from the Latin word fervere. So fervere means simple meaning is to boil. Whenever we take an liquid, in the liquid we put an microorganisms. So after the doing their metabolic activity, microorganisms releases carbon dioxide gas, and when the carbon dioxide gas bubbles comes on the surface of that liquid, which is known as fervor. So after the modernization, the term fervor converted into the fermentation. So then the scientists use that fermentation activity of that particular microorganisms, because scientists already knows that microorganisms after the metabolic activity produces and different types of products just like an organic acids, alcohol, but in small scale it is not much to use, but at a large scale they are using it. So for the like modern definition, they state modern definition. So any controlled large scale cultivation of microorganisms or their enzymes in a bioreactor to produce commercially valuable products regardless of whether the metabolism is strictly anaerobic. So, so large scale cultivation means up to 10,000 liter to 5 lakh liter. At that much level, at this much large scale, the microorganisms are used for the fermentation to formation of desired products, just like at a large scale of alcohols and different types of enzymes, antibiotics. So all these reactions and all these process takes place in a controlled manner. Controlled manner means the different types of parameters just like temperature, pH, osmotic pressure, viscosity, all that parameters in a controlled manner, and these processes takes place in a vessel which is called as bioreactor. So all of the bioreactors are fermenter, but not all the fermenters are the bioreactor. So this is a simple definition. So in a bioreactor to produce commercially valuable products, all by the industrialization, all this fermentation reaction produce and industrially important products because of the fermentation processes, and these commercially valuable products be in a market to sell. So we are moving forward. So we are moving forward to the history of fermentation. So before thousands of years ago, humans have used fermentation for the formation of different types of food products, but they don't know about it. So ancient origins said that humans have used fermentation unknowingly for thousands of years. But after, when the Louis Pasteur comes in the year of 1850s, he proved that fermentation is caused by living microorganisms. It means ferments, and he also states that it is distinguish it from putrefaction. So fermentation term is a different things and putrefaction is totally different. So, and he also gives pasteurization. So what is meant by pasteurization? Pasteurization is a heat treatment process that destroys harmful pathogens in foods and beverages to prevent spoilage and illness. So in the scientific understanding, the fermentation is to ferment the product and the formation of different types of useful product and also release of carbon dioxide. And what is meant by putrefaction? Putrefaction is to decompose dead matter and decomposed to and decomposed protein which is present in muscle cells in the dead animals. So, and the third term is the industrial revolution. So after learning ancient origins and scientific understanding which is given by Louis Pasteur, we are moving forward to industrial revolution. So in industrial revolution in early 20th century saw large scale production of acetone, butanol, citric acid and later antibiotics. So for the industrial, all of we know microorganisms form by the metabolic process, they are forming different types of useful products. So acetone, butanol and citric acid, just like citric acid is very helpful in a cosmetic industry, but at this much scale, at very large scale, it is impossible to it extract from the fruits. So we are using microorganism to form it at a large scale. So and antibiotics, just recently we are producing antibiotics with the help of fermentation processes, and the first of all penicillins, which is formed because of flora and chain in the year of 1940s. So now we are moving forward to next topic. So we are continuously saying metabolic pathway, metabolic pathway. So what is my metabolic pathway? So glycolysis is the first metabolic pathway which is microorganisms use after the taking of nutrients. So by the taking of nutrients, glucose is entering the cell, and the cell is getting broken down the glucose into the pyruvate and energy molecule and different types of other products, and all of this glycolysis process is occurring in a cytosol of cells and yields net of 2 ATP and 2 NADH. 2 ATP and NADH are the energy molecules, and here we are saying 2 ATP and 2 NADH molecules as per glucose molecule. It simply states that one glucose molecules which is broken down into pyruvate, which gives two molecules of ATP and 2 NADH. It's like simple calculation. When the two glucose molecules, it's like 4 ATP and 4 NADH. So what we are going to learn, glycolysis. So glycolysis is a 10-step process. So simply mnemonic term that we are going to learn is we made a sentence like good girlfriend fill glass daily by 3 pg and 2 pg play ping pong. Good simply states that glucose, it means a 6 carbon sugar, and then glucose, and it get converted into the glucose 6 phosphate, it gets the phosphate type. Then glucose 6 phosphate converted into the fructose 6 phosphate, which is changes their shape, and that fructose 6 phosphate converted into the fructose 1,6 biphosphate. It gets another phosphate tag. So after it, which is converted into the glyceraldehyde 3 phosphate, the 6 carbon sugar splits into 3 carbon sugars, and then it get converted into the dihydroxyacetone phosphate. After it also get one another phosphate tag and converted into the 1,3-biphosphoglycerate. After it 1,3-biphosphoglycerate converted into the 3-phosphoglycerate and 2-phosphoglycerate, which is 3-phosphoglycerate also known as 3-PG and 2-phosphoglycerate also known as 2-PG. After it get converted into the phosphoenol pyruvate also known as PEP, and after the removing of phosphate group, only the final product which is we are getting is a pyruvate. So we are moving forward. So the glycolysis is a metabolic pathway which is a 10-step process. We are simply converted into the two phase, energy investment phase and energy payoff phase. So in the first of all energy investment phase, we are investing energy in the different form, and then in the second phase, energy payoff phase, we are getting an energy in a different form. So glucose in an energy investment phase, glucose with 2 NAD plus molecule with 2 ADP plus and with the 2 molecules of phosphate, which ultimately gives an 2 pyruvate, 2 NADH plus 2 hydrogen ions and 2 ATP molecules with the 2 molecules of water. So NADH and ATP, these are the main energy molecules which is required for the electron transport chain and a different cellular activity. So these are the simpler classification. So depending upon the microorganisms, parameters, and also depends on different types of nutrient media. There are different fermentation types regenerate NAD plus molecules. So there are main fermentation types are alcoholic fermentation, lactic acid fermentation and mixed acid fermentation. So first of all, alcoholic fermentation. In this case, in this fermentation typically performed by yeast, means alcohol as a product which is formed by yeast, which is also known as Saccharomyces cerevisiae. It is a scientific term for the yeast, which in this process converts sugar molecule into ethanol and carbon dioxide. So simply glucose, which is molecular formula is C6H12O6, converted into 2C2H5OH, which is known as ethyl alcohol or ethanol, and 2 molecules of carbon dioxide and energy. So in this case, in this reaction pattern, we are getting our alcohol or ethanol by cellular activity or microorganism perform their activity. So after the alcoholic fermentation, we are moving forward to lactic acid fermentation. So in this fermentation performed by bacteria, just like in alcoholic fermentation it is because of yeast which is a fungus, in the lactic acid fermentation it is because of bacteria and animal muscle cells, which is in this process it get converts sugar into lactic acid, means glucose, which is also known as molecular formula is C6H12O6, which is directly converted into the 2C3H6O3 and energy molecule. So it is because of different types of bacteria, just like lactic acid bacteria, lactococcus, lactobacilli, just like it. So we are now moving forward to the mixed acid fermentation. So in the mixed acid fermentation, mixed acid fermentation is a different types. In this action is performed by mainly enterobacteria. It means enterobacteria performs mixed acid fermentation process. But in this case, not by glucose we are getting mixed acid, but because of by glycolysis process which is converted and glucose into the pyruvate which is the product, we are using that pyruvate for the formation of different acids. So pyruvate get converted into the acetyl coenzyme A and formic acid, and this pyruvate converted into the acetyl coenzyme and formic acid by what? It is because of the Krebs cycle or citric acid cycle. So after the formation of this acetyl coenzyme A or formic acid, and after it's totally depending on bacterial strain, different types of bacterial strain, which formic acid get broken down into different gases by enzyme formate hydrogenylase. So in the mixed acid fermentation, different types of products and multiple products are being formed, just like lactic acid, acetic acid and succinic acid. So we are done here by the introduction of the fermentation process. So in this image, all of you can see that glucose is entering into the cell, and by the glycolysis, glucose is converted into the pyruvate, and after this, the pyruvate converted into the acetaldehyde because of the enzyme pyruvate decarboxylase, and the acetaldehyde get converted into lactate uh, because of the lactate dehydrogenase. It is a first step, it is a single chain, but in the second chain, by the green arrow, pyruvate get converted into the phosphoenol pyruvate, and then the phosphoenol pyruvate get converted into the oxaloacetate. It is because of P carboxylase enzyme, and after this, oxaloacetate converted into the malate, malate get converted into the fumarate, and fumarate converted get into the succinate. So it is because of the different types of enzymes, different types of bacterial strain, because in the different types of bacterial strains having and different types of enzymes, so it is because of that different types of acid are being formed. So here we are done by the introduction of fermentation process. Thank you all of you for carefully listening this video lecture.